This is the first in a series of 3 podcasts on epithelium. Epithelium is the first of the 4 basic tissue types that we're going to study in our course. Obviously there's epithelium, there's connective tissue, there's muscle, and there's nerve. And of course these 4 basic tissue types come together to form all of the organs in the body. Here are your learning objectives for these series of podcasts on epithelium. Would like you to be able to understand basic structure and function of epithelium. You should be familiar with the various classifications of epithelium and where epithelia are located in the body. Would like you to understand something about the structure and function of basal lamina and of junctional complexes between epithelial cells. Would like you to appreciate the structural and functional polarity of epithelial cells, and would like you to understand some of the structure and function of surface appendages on epithelial cells. Now for the laboratory, you should be able to recognize various types of epithelium, not only from electronic images, but also on glass slides. Now there are several basic concepts we'd like you to keep in mind as we talk about epithelium. Epithelium can form a continuous, uninterrupted sheet of cells, so you could say that epithelium would cover the body surfaces, would line body cavities, and would constitute glands. Epithelium would separate the internal milieu of the body from the external environment, so epithelium can invaginate, it can proliferate, and of course it can specialize. Epithelium plays an important role in homeostasis, so it can regulate the content of the internal milieu, not only of individual organs, but of the body at large. Epithelium can function as a barrier, so it can be involved in selective transport across a surface of the body. It can function in various transport capabilities and in permeability. So this simple-minded cartoon that we've drawn, imagine a cell dividing and forming, in this case, a ball of cells. But let's just say this ball of cells represents an organ or even an entire organism. You might imagine that surface cells might specialize and perform a different function than the cells internal to the organ. You might imagine, for example, these cells on the surface might function to prevent transport of material across the surface, or the epithelium might function to allow material to cross the surface in either direction, so it could allow material to move into or out of an organ. 3 principal characteristics of epithelium. They're closely opposed to and adhere to one another by means of specific cell-to-cell adhesion molecules. These molecules can form specialized junctions, and we will look at these in a little bit. Epithelium can exhibit a functional and morphologic polarity, so different functions can be associated with 3 distinct surface domains on epithelium. We're going to come to talk about an apical domain or a free surface domain on epithelium. We'll talk about a lateral domain, that would be a cell-to-cell surface domain on epithelium, and we'll talk about a basal domain, that would be the area of epithelium that might rest on an underlying basement membrane or basal lamina. Basal surface is usually attached to an underlying basement membrane. We'll talk about this in some detail later, and epithelial cells will contribute to the synthesis of basal lamina, as will cells of underlying connective tissue. In special situations, epithelial cells lack a free surface. We might talk about, for example, epithelioid tissue. These are epithelial cells that lack a free surface, and for example, endocrine glands don't have a free surface necessarily. They secrete into the blood system. The thymus is another example of an epithelioid type tissue where there's not a free surface. Some common features of all epithelia. Epithelia are highly cellular. There may be many identical cells in an epithelial sheet, but with little extracellular matrix materials. Epithelia are almost always avascular, that is, they don't have a blood supply, so to get nutrients there has to be diffusion. Now, there are a couple of exceptions to this, notably the stria vascularis in the inner ear is an exception to this, and we will talk about this epithelium much later in the course. Epithelium may have a nervous innervation, so there'll be sensory portions to epithelium. Epithelium divide fairly rapidly, so they have a high mitotic index, reflecting a fairly rapid cell cycle time. Epithelium rest on a basement membrane, as we alluded to just a minute ago. Most epithelia lie on a basement membrane, and epithelia contribute to the synthesis of this basement membrane, as do the underlying connective tissue cells. There are junctional complexes between epithelia to attach epithelial cells cell to cell, and maybe also to help attach epithelium to their underlying basement membrane. And epithelia are polarized, and this polarization reflects structure and function, so we often say that the structure reflects the function. As we'll come to understand, epithelia can be polarized for transport, and you might already have a feeling that epithelia may be polarized for processes like endocytosis and exocytosis. Now, every surface of the body can be covered by an unbroken sheet of epithelium. So, for example, you might think of the skin, the epidermis, an epithelium that's lining the outer surface of the body. So every surface of the body covered by an epithelium. But as you think of the GI system, there's an epithelium lining the surfaces of the GI system. So every surface of the body lining by an epithelium, and this epithelium tends to be an unbroken sheet of cells. So the epidermis would be an unbroken sheet of cells. You know that if you abrade or have a break in the epithelial surface, the body will heal itself. Unbroken sheet of epithelium along the GI tract. Likewise, unbroken sheet of epithelium lining the respiratory tract. So here I'm tracing it, the nasal cavity, for example, coming down the pharynx, into the larynx, the trachea, and even going into the lungs to the very smallest alveolar surfaces of the lungs. There's an unbroken sheet of epithelium. Because you might imagine that as you're going down into the very smallest surface of the lungs, or if you go back to this diagram going into the GI tract, you're actually within these organs on the outside of the body. Likewise, there's an unbroken sheet of epithelial cell that's lining the cardiovascular system. So, for example, in the chambers of the heart or in the blood vessels, an unbroken sheet of epithelium. We talk about mesothelium as an epithelium that lines body cavities. So, for example, the visceral pleura, the parietal pleura, the pericardium, the peritoneal cavity, that epithelium would be considered a mesothelium. When we talk about the epithelium lining the cardiovascular system, we talk about an endothelium. So we talk about the epithelium on the surface of the body as the epidermis. Now, epithelia are derived from all of the germ layers. This is a fairly complicated slide. We can make it a little simpler by looking at it on this slide. Epithelium of the skin and associated glands, oral and nasal mucosa, epithelium in the cornea, mammary glands. That's all epithelium derived from the ectoderm. We also know that you have neuroectoderm, so neural crest cells, for example, give rise to adrenal medulla and melanocytes. From the neural tube, you have the retina, the pineal, posterior pituitary, and the ependymal cells lining the central nervous system. From the endoderm, the epithelium of the liver and pancreas, or the epithelium lining the GI tract and respiratory tracts, would come from endoderm. From the mesoderm, linings of the urogenital tract, so like kidney tubules, reproductive tracts, the endothelial lining of the cardiovascular system, or the mesothelial lining of the body cavities would all be derived from the mesoderm. So, just to make the point, epithelium derived from all of the germ layers in the embryo. Basic functions of epithelium, as you might imagine: protection of underlying tissue from abrasion or from injury. You might imagine transcellular transport of molecules across epithelial layers. Epithelium is involved in secretion, so for example secretion of mucus, of hormones, or of various enzymes. Epithelium plays a role in absorption of material from a lumen, so you can appreciate, for example, absorption of material in the intestinal tract, certainly absorption of material in tubules of the kidney. Control of movement by selective permeability, and this will be because of the junctional complexes between epithelial cells. And you can imagine epithelium detects sensations. So for example, there's taste buds in the oral cavity. Epithelium that make up the retina clearly detect sensations. The epithelium that make up the hair cells in the inner ear clearly detect sensations. So I think you can get an appreciation of the sensory function of epithelium. You can classify epithelium based on the cell shape. So we talk about a squamous epithelium, that is a flat cell shape. We talk about a cuboidal epithelium, where the cell is about as tall as it is wide. So like a little bit of a cube. We talk about a columnar epithelium, where the cell is substantially taller than it is wide. And we'll also talk about a transitional epithelium, which is relatively unique to the urinary system, and that epithelium actually has the ability to change shape somewhat, where you can go from almost a very flat, almost squamous epithelium to a relatively high cuboidal epithelium, and we'll show you some examples of that in a little bit. We can also define epithelium by the number of cell layers. So we can talk about a simple epithelium or a stratified epithelium. And here we're going to define the epithelium by the shape of the surface layer of cells. So we can also talk about a pseudo-stratified epithelium, and we'll come back and talk about that in a few minutes. So not only can you define epithelium by the cell shape, but you can define epithelium by the number of cell layers in the epithelium. And we can also talk about epithelium by the types of specializations present. So for example, would there be keratin on the epithelium? Would the epithelium have a cilia on the surface? Or would there be microvilli or stereocilia on the surface? And so let's look at classifications of epithelium. Major classifications: we'll talk about a simple epithelium, a stratified epithelium, a pseudostratified epithelium, and we'll talk about a transitional epithelium. So in the diagram, we can talk about a simple squamous epithelium, a simple cuboidal epithelium, or a simple columnar epithelium. Notice the simple squamous epithelium is a single layer of flat cells. The simple cuboidal epithelium, a single layer of cells that are about as tall as it is wide. And the simple cuboidal epithelium, a simple layer of cells, a single layer, but the cells are much taller than they are wide. So simple squamous epithelium, for example, the lining of Bowman's capsule in the kidney, the alveoli in the lungs would be a simple squamous epithelium. The mesothelial lining of the body cavity is a simple squamous epithelium. The endothelial lining of blood vessels or lymphatic vessels is an endothelium, but it's a simple squamous epithelium. So if we're talking about the lining of body cavities, we give the simple squamous epithelium, we give it the name mesothelium. If we're talking about the lining of blood vessels, we give that simple squamous epithelium the name endothelium. So these terms, mesothelium, endothelium, just let you understand or know that you're talking about a specific region of the body. The epithelium lining the small exocrine ducts of glands might be a cuboidal epithelium. The germinal epithelium on the ovary would be a simple cuboidal epithelium. Epithelium lining kidney tubules or thyroid follicles would be a simple cuboidal epithelium. On the other hand, a simple columnar epithelium, you would see that, for example, lining the GI tract or in the gallbladder. If we talk about stratified epithelium, we can talk about a stratified squamous epithelium lining the oral cavity, the esophagus, or the vagina. We can talk about a stratified squamous epithelium on the epidermis of the skin. Here we'd be talking about a stratified squamous epithelium with keratin. So we might, for example, say keratinized stratified epithelium. If we talk about a stratified cuboidal epithelium, we might be talking about the epithelium that makes up sweat glands or that lines large exocrine ducts. If we talk about a stratified columnar epithelium, we might be talking about epithelium lining the largest exocrine ducts. So here we've got a stratified squamous epithelium, non-keratinized. So as we said, like in the oral cavity or the esophagus or the vagina. A stratified squamous epithelium, keratinized, like on the surface of the skin. And then, as we just mentioned, stratified cuboidal, stratified columnar. If we talk about a pseudo-stratified columnar epithelium, we'd be talking about epithelium lining the trachea or the bronchial tree, maybe some of the organs in the male reproductive tract, like the vas deferens, the epididymis. It's a pseudo-stratified columnar epithelium. It appears that the epithelium is in multiple layers, like here you have a stratified columnar epithelium, multiple layers of epithelium, but every cell actually is going to touch the basal lamina. That's why we would talk about it as a pseudo-stratified columnar epithelium. Now, let me get back for a minute and talk about this stratified epithelium. Here, we're talking about a stratified squamous epithelium. We're defining this as stratified squamous because the very top layer of cells is squamous or flat. A stratified cuboidal epithelium, the top layer is cuboidal. A stratified columnar epithelium, the top layer is columnar. And the diagram makes this point really well. Here, if you looked in this area, you might be tempted to say they're cuboidal cells, but we're defining the epithelium here as a stratified columnar because the top layer is columnar. When we talk about transitional epithelium, we're talking about an epithelium basically that's in the urinary system. So sometimes you'll see the term urothelium. This would be epithelium lining the renal calyces, the ureter, the bladder, the urethra. And what happens in this transitional epithelium, the surface cells can become very highly distended, almost squamous, or they can bunch up and become almost cuboidal. So we call this a transitional epithelium because the surface layer of cells has the ability to change shape. If the bladder is very highly distended because it's full, the surface epithelium would be very flat. On the other hand, if the bladder has just been emptied, the transitional epithelium is relaxed and the surface cells would be almost like a cuboidal shape. So transitional epithelium has the ability to change shape somewhat with respect to the surface cells. Here is a table that I'd like you to look at in some detail to look at the various classifications of epithelium. I'm not going to go over it all. You can read this from your notes, but remember that you can classify epithelium by the shape of the surface cell, whether it's flattened, so squamous, whether it's cuboidal, or whether it's columnar. You can talk about epithelium as pseudostratified. All cells, as we said, rest on a basal lamina, but all cells don't necessarily reach the surface. Usually you see a pseudostratified columnar epithelium. Now, we will talk a little bit later about pseudostratified columnar epithelium being ciliated versus non-ciliated. We talk about stratified squamous, and we can talk about whether the epithelium are keratinized or not. The epithelium are keratinized, that would reflect the epithelium on the surface of the skin. The very surface cells are very highly flattened. They lose the nuclei, and the surface layer of cells actually dies, but the surface layer of cells provides some amount of protection for the epithelium for the epithelial cells that underlie it. Transitional epithelium, where the surface cells are relaxed, almost a cuboidal shape. They're very much like a dome-shaped cells, and we've talked about where you find these cells. There can be various specialized cells within epithelium, and some of the epithelial cells themselves can specialize. So, for example, goblet cells are specialized epithelium that play a role in secretion. APUD cells are specialized epithelial cells in certain organs that can secrete hormones. Now, you may have lymphocytes that are a specialized connective tissue type of cell that can infiltrate an epithelium. You can have an antigen-presenting cell that can infiltrate an epithelium. These cells function in immune activities of the body. You can have sensory epithelial cells like nerve cells that are involved with epithelium. These cells receive input in the body. There can be a variety of epithelial cells in an individual organ. So this slide shows a section in the medulla of the kidney, and there are several types of epithelium that are evident on this slide. First of all, there's a simple squamous epithelium that lines the thin limbs of Henle's loop, and that's shown by these arrows. Thin limbs of Henle's loop lined by a simple squamous epithelium. On the other hand, a simple cuboidal epithelium, like shown here or shown at these arrowheads, would line some of the collecting ducts in the kidney, and a endothelium shown here would line the vasorecta. And we will talk about these different areas in the kidney as we talk about the kidney much later in the course. But a very simple squamous epithelium, but this vasorecta is a blood vessel. You can see red cells in the lumen of these vessels, so the epithelium here would be given the name endothelium. Here's the renal cortex in the kidney, showing a renal corpuscle, simple squamous epithelium lining the parietal layer and visceral layer of Bowman's capsule. Here you can see on the parietal layer the very flat epithelium, the simple squamous epithelium here. More difficult to discern that on the visceral layer of Bowman's capsule. But we'll talk about that later in the course. Here on this magnification, you can see the simple squamous epithelium on the parietal layer. More difficult to pick out the simple squamous lining on the visceral layer here, but we'll get to that later in the course. Small intestine, simple columnar epithelium here or here. Here's a goblet cell where I've got my pointer. This would be the luminal surface. So you can see these goblet cells. You can also see some lymphocytes that are infiltrating this simple epithelium. The surface is specialized into a microvillus brush border, and in this crude diagram that I've shown, here would be 3 of these simple columnar epithelial cells, nuclei shown here. Here, the brush border, the surface is thrown into folds, and these folds, as you might imagine, increase membrane surface area. And here we've just shown these cells resting on a basal lamina. Here's the esophagus, stratified squamous epithelium lining the esophagus. Note we're classifying this because the surface layer of cells is squamous or flat. If you'd looked at some of these cells deeper in, you might be tempted to think that's a cuboidal type of cells, but we're defining it by the surface layer of cells. Here's thick skin, a stratified squamous epithelium with keratin, and this entire layer of cells is essentially a dead layer of cells. So the living cells are here on the surface. It's a stratified squamous. It's multiple layers of cells, so it's stratified. It's squamous because the surface cells are flattened, but it's a stratified squamous with keratin because of this pretty substantial keratinized or dead layer of cells on the surface. Here's the dermis of the skin, and we can talk about a stratified cuboidal epithelium on the duct portion of a sweat gland. It's stratified because you can see two layers of epithelial cells. The secretory portion of the gland is a simple cuboidal epithelium. This is deep down in the dermis, the junction between the dermis and hypodermis. This is areas of connective tissue, and we'll get to that later in the course. Right now I just would like you to recognize the simple cuboidal epithelium on the secretory portion versus the stratified cuboidal on the duct portion of this gland. This stratified epithelium on a sweat gland is sometimes given the name double cuboidal epithelium. Now at this point, don't worry that you can't necessarily identify this as a sweat gland or that you can't identify this as the dermis and the skin. The course will build on itself, and this will become clearer as you begin to look at more and more of the material in the laboratory. This is an example of the urinary bladder, this transitional epithelium, showing these characteristic dome cells on the surface, on the luminal surface. So this would be the luminal surface. This is where the urine would be coming in contact with this epithelium. We can also define a lamina propria. The lamina propria of the bladder, there's a superficial dense, irregular connective tissue and a deeper, loose connective tissue. So this area is more dense, this area is less dense. And when we talk about a lamina propria, this is going to be a term that you hear repeatedly in the course. Lamina propria is the underlying connective tissue upon which an epithelium rests. As you'll come to understand, most epithelium does rest on a connective tissue or lamina propria. Here's a view of the trachea with a ciliated pseudostratified columnar epithelium. This ciliated pseudostratified columnar epithelium may be given the name respiratory epithelium because it's the only area in the body where you find ciliated pseudostratified columnar epithelium or in the respiratory tract. This is pseudostratified columnar because it looks like there are multiple layers of cells, but if you could trace these cells, all of these cells rest on a basement membrane, but not all of the cells necessarily reach the free surface. So it's a pseudostratified columnar epithelium. Epithelial cells have polarity. There's an apical domain, which is always directed to the exterior surface or luminal surface if we're talking about an enclosed cavity or tube. There's a lateral domain where the cells contact and communicate with each other. There may be specialized attachments or junctional complexes between adjacent epithelial cells. And then there's a basal domain where the cells rest on a basement membrane, and there may be specialized structures that anchor these cells to the underlying connective tissue. We're going to talk about the apical domain in epithelium first. The apical domain will have specialized appendages on the surface. So, for example, microvilli on the surface will increase membrane surface area. There may be cilia on the surface that will play a role for movement. There may be stereocilia, which would increase membrane surface area. Here is an electron micrograph showing microvilli on the surface, higher power view. There's an actin core in these microvilli. Here you can see it by freeze fracture very nicely, an actin core, but the microvillus brush border is very uniform, and of course they're covered by a plasma membrane. This is a light micrograph in the GI tract. You can barely get a sense of this microvillus brush border, but there's a glycocalyx adsorbed onto this microvillus brush border. This glycocalyx stains positively with periodic acid Schiff. So that's why you see this intense red staining near the surface. And then the goblet cells secrete material that makes up this glycocalyx, so the goblet cells are staining PAS positive. Go back and you can see the microvilli this way. Here is a microvillus brush border, maybe like in a hepatocyte, a much less intense or much less organized microvillus brush border, but you can still get a sense that there are some surface appendages coming off of this surface of the cell. So what you can say clearly is the microvilli are increasing the membrane surface area. This just is what we've just said. The microvilli have actin cores, just showing this diagrammatically here. The actin cores are cytoskeletal components. So you've got actin in the terminal web of the cell, actin going into the microvilli themselves, and then other cytoskeletal proteins can interact with these actin. So we can say the terminal web contains actin, it contains myosin, spectrin, and a variety of other cytoskeletal proteins, and then these cytoskeletal proteins in the terminal web will interact with intermediate filament proteins, various cytokeratins, and we'll talk about in more detail as we talk about cytoskeleton. This is an example of stereocilia. Stereocilia are just essentially very long microvilli. They're sometimes branched. They also function to increase membrane surface area. Stereocilia are non-motile, so be careful. Don't think that because we're saying stereocilia they're motile. As we'll talk about, cilia are motile structures, but microvilli and stereocilia are non-motile structures. They have actin cores, whereas cilia have a core of microtubules. And that gets us to this right now. Cilia or flagella have a microtubule core, an axoneme consisting of a core of 9 microtubules, 9 plus 2 actually. The microtubules are covered by a plasma membrane. At the base of the ciliary axoneme is a basal body. It's basically 9 triplet configurations of microtubules. This we'll talk about in more detail later in the course. But these microtubules are motile. So here on this pseudostratified columnar epithelium in the respiratory tract, these microtubules actually can move. So the cilia beat, and you might think these beating cilia serve a function in the respiratory tract, for example, to move material in the respiratory tract. This at the electron microscopic level shows microtubules going up into the core of these cilia. Here's a very nice cross section showing the very characteristic 9 plus 2 arrangement of microtubules, as you might see in the diagram here. The basal body showing 9 triplet microtubules without the central doublet. This will be talked about in much more detail later when we talk about cytoskeleton.